Last time we left off with the introduction of what metabolism is, right? So, metabolism is the sum of all the chemical reactions in the body. A metabolic pathway is a sequence, specifically step by step, which molecule gets synthesized by which enzyme, or which molecule gets broken apart by which enzyme, right? And remember, at each time there's an enzyme that's very specific for its substrate, and there will be a rearrangement of the configuration of the electrons, which leads to breaking a bond or making a new bond. Okay. Of course, all of the enzymes in an organism are encoded by genes. And we also introduced the idea that metabolic pathways are coupled because one pathway will be like anabolic, which uses energy, and then another pathway will be catabolic, which releases energy. So as soon as that ATP gets made by catabolism, it's going to get used by an anabolic process. Okay, cells do not store ATP. If a cell has enough energy, it will slow down the production of ATP, and if it needs to store up extra energy because it has more than it needs, then it's going to store up glycogen. And once all the glycogen reserves are full, then it's going to put everything else into fat. Okay? So this is demonstrating the coupling, right? So as glucose is broken down over many steps, it's going to release ATP throughout the process. As soon as that ATP gets made, it's going to be used up by some anabolic process that needs the energy. In this example, it's taking amino acids and making proteins. Sometimes it's actually synthesizing the specific amino acids that we need, right? It could be making a different type of carbohydrate that we need. Any molecule and any process that the cell needs to carry out. Okay? And also, I want you to remember that oxidation means the loss of electrons, so it's becoming more positive, and a reduction means that it's gaining electrons. Electrons are negative, so it's becoming more negative. Okay, and so a lot of times we'll see redox reactions in metabolism as something that had a plus sign that no longer has a plus sign, and that's the reduction, right? So FAD plus becomes FADH. It lost the plus sign. It's no longer positive. It became more negative. It gained electrons from the hydrogen that it picked up. Okay? So energy production, energy associated with the electrons that form bonds between the atoms in a molecule. Cells concentrate this energy, this chemical energy in bonds. It concentrates this in phosphate bonds, right? ATP, it stands for adenosine triphosphate. Those phosphates are highly electronegative. They have a lot of electrons, and they do not like to be next to another highly electronegative atom, such as itself or oxygen, right? And so putting 3 phosphates together in a row is not stable, and it's like bursting with energy, waiting for that 3rd phosphate group to pop off, releases energy that can be used to do work, whatever that work is that the cell needs to do, making some new monomer, making a new, putting monomers together to make polymer, right? Anything that it needs to do. Okay? So the cell can generate ATP, can put on that 3rd phosphate group, which we call phosphorylation. So you phosphorylate something, you're adding a phosphate group to it. So when we phosphorylate ADP, which would be D for diphosphate, when we phosphorylate that, then it becomes ATP, triphosphate, okay? And we can do this in 3 ways. Or we cannot actually do the third way. We have 2 ways in our body that we can do this. There exists in nature 3 ways. Okay, so the first way is what we call substrate level. So it's taking the phosphate directly from one molecule and putting it onto the ADP. And this happens during what we call glycolysis, which we'll see a lot today. Okay, and then we have oxidative phosphorylation, where you carry out redox reactions. You'll have to have a final electron acceptor to end those redox reactions. And you harness energy from each of those redox reactions to do this work. So we see this in the electron transport chain. And so we'll look at that in a little while. And then the third one, which we don't do in our body, but is, prevalent in nature, would be, in photosynthesis, is using the light. So photophosphorylation. And we will not even look at photosynthesis in this class. We don't have time. I know that general bio will have a huge focus on photosynthesis. 4A also will have a section on photosynthesis, so I do not feel the need to cover it in our class, although we do have some photosynthetic microbes. Our photosynthetic microbes are not pathogenic, so I'm choosing to, I can't cover everything in one class, right? I have to cut something somewhere. So that's one of them. All right, so energy production and catabolic reactions. We'll start by looking at carbohydrate catabolism, and we'll see how other types of so we'll look at a few types of carbohydrate catabolism, and then we'll see how other foods like lipids and proteins, how those are broken down as well. Okay. So as I've already said, metabolism is the sum of all chemical reactions. Anabolic reactions require ATP for building things, okay? Catabolic reactions will supply that ATP. And also, catabolic reactions are giving you the raw materials that you need as well. So supplying amino acids and carbohydrates that you can modify into whatever type you need. And then you can once you have all the monomers that you need, you can put them together into whatever molecule you're trying to make. So where do we get the energy and the building blocks? Of course, from the food that you eat, the different carbohydrates, lipids, and proteins that you're eating. So recall that you eat, it gets into your small intestine, and you start to break it down and absorb it. And this is just showing glucose, but we still absorb protein and fats through our gut as well. They have slightly, they'll have slightly different pathways once we get to the specifics of how they get absorbed and the path that they travel, and then the specifics of their metabolic pathways, right? But in general, this is just a good image to help you remember that you eat, it goes into your bloodstream. From your bloodstream, it then can go into your tissues, and then this is showing going into an individual cell, getting into the cell, and then once it's into a cell, it can begin being broken down, and so we see glycolysis. If you have this is us, right? This is a person. So if you have oxygen present, it's going to go all the way through the full set of pathways called cellular respiration. If you are working out really hard, or if you're incredibly ill and you don't have enough oxygen, then you're going to go to this pathway to the right called lactic acid fermentation. Okay, so lactic acid fermentation you're familiar with when you work out and you get sore, you feel the burn in your muscles, and then for the next day or two you're achy. That is from the lactic acid buildup. Why does the achiness get better over time? Like it'll become, the second day you're less achy, and then the third day it's gone. That's because we will actually, once the oxygen becomes available again, we start turning that lactic acid actually back into pyruvic acid so that we can feed it back into the system and get the rest of the energy out of that molecule. And so it takes time for our body to absorb the lactic acid that we made and process it and use it. And so that's really what I'm showing here. And so in this space, glycolysis is within the cytoplasm or cytosol of the cell. And then it moves into, once glycolysis is finished and we change pyruvic acid into acetyl-CoA, that goes into the citric acid cycle, which I like to call the Krebs cycle, right? And that's inside of the mitochondria, and this is inside the matrix of the mitochondria. Then you go to the electron transport chain, and that goes into the inner membrane of the mitochondria, and that's where you complete and get the vast majority of the energy. Okay, so this is a nice visual. Lots of different ways of looking at things, so take time to understand each of the images, because it challenges your mind to see how well you actually understand it. I've said this before and I'll say it again. It's much easier to follow along when someone's explaining something than it is to do it yourself. So a really good study habit is to try explaining concepts, explain diagrams, right? All these different things. Explain them out loud so you can't trick yourself into thinking you know it. Some of us are really good at constantly questioning and making sure that we actually understand what we're looking at and what we're reading. And some of us are not good at catching where we don't understand something. So the more you practice explaining things out loud, the better you're going to become at identifying your areas of weakness and improving upon that. Okay, so cellular respiration is a catabolic pathway to generate ATP. Glucose is going to be oxidized. Molecules throughout the pathway are going to be oxidized, and then others are going to be reduced, right? So there's going to be redox reactions, and you will have to have a final electron acceptor. And that final electron acceptor will come from outside of the cell, and it will be inorganic. For us, it's oxygen. For a lot of microbes, it's also oxygen. But we do have a subset of microbes that can use an alternative electron acceptor. And so because it's not using oxygen in that case, we call it anaerobic respiration. And we'll talk about that a little bit at the end once we get through this. Okay, so what are the pathways of cellular respiration when we're we'll go through these processes focused on having oxygen. Okay, so you will start with glycolysis to take one molecule of glucose and break it apart and get the process started. You'll end with what we call pyruvic acid, which will be modified in the intermediate step to acetyl-CoA. Acetyl-CoA then feeds into the Krebs cycle. This is happening within the mitochondria now. The Krebs cycle gives you a lot of what we call electron acceptors, which are essential for the electron transport chain. So they transfer electrons to the electron transport chain, which then gives you the biggest series of redox reactions to get the most energy from that glucose and transfer it into ATP. So we'll see how that all works. And then, as I already mentioned, if the final electron acceptor is not oxygen, then it's called anaerobic. If the final electron is oxygen, it's called aerobic. Okay. Sometimes instead of clarifying, we just say cellular respiration, and by default we mean aerobic cellular respiration, unless otherwise clarified. Okay. And you have to remember, anaerobic respiration is still respiration. You're still going through all of these steps. It's not going to have oxygen at the end of the chain. And because of that, it actually won't be able to get quite as much energy as what we're going to be looking at with aerobic. Okay. This doesn't tell you whether or not, like, when you see an anaerobic process, right? When an organism is anaerobic, it has something other than oxygen as its final electron acceptor. That doesn't tell you whether or not it can tolerate oxygen. It doesn't tell you whether or not it can switch between the two. There's a lot of complex. Each organism, especially when we're talking about microbes, is unique. So the specific type of molecules that it's using, the environments it can survive, and the environments it prefers, right? These are all different for each organism. And because of that, we can use these different characteristics to identify an unknown bacteria. And so that's why we're studying all of this, right? Okay, so the equation for aerobic cellular respiration is C6H12O6, which you recognize as glucose, oxygen, okay? And then you get out of this some waste products of water and carbon dioxide and heat, right? And then the goal product is the 32 ATP. This number is taking into account the loss in a person. We have to expend some energy to move molecules from the cytoplasm to the mitochondria and such. So you're going to see numbers that add up to more than 32. So this is just saying, like, the actual yield that we observe in eukaryotic cells, especially people. Okay. And then we'll go through each process looking at its actual isolated amounts, so not taking into account any losses. Okay, so glycolysis, as I mentioned, is taking a glucose into the cytosol, right? So glucose has to get transported across the membrane through a channel, right? And then you will split it in half. Okay, so glucose is a 6-carbon sugar, and you're going to split it in half, and you'll end up with 2 pyruvic acid molecules, which each have 3 carbon molecules. Okay, there are actually many steps involved, 10 different enzymatic steps in order to get from glucose to pyruvic acid. Okay, I'm not asking you to memorize those individual steps. I'm asking you to memorize what goes in and what comes out. Okay, so you put in 1 glucose and you actually have to invest 2 ATP to get the process started. You are going to send 2 NAD plus molecules as well, and you will get out the 2 pyruvic acid molecules. And at this point, you get out 4 ATP, but remember you invested 2 of them, and this NAD plus will pick up hydrogen and will become NADH. Okay, so at this point, you have made 2 ATP, but you have these molecules, these pyruvic acid molecules, that still have a lot of energy in their bonds. So we could oxidize them further and get more energy from them. And so we do this by modifying it and putting it into the Krebs cycle. And then the 2 NADH are holding some energy that we've taken from glucose already, and they're going to go to the electron transport chain to harness that energy. Okay, so we have made 2 ATP, so we already made a little bit of energy from glucose, but we have a lot of potential energy stored in the pyruvic acid molecules and the NADH molecules. Okay, and that's why it's so important that we are able to do the Krebs cycle in the electron transport chain so that we can get all of that potential energy. Really, glycolysis does not require oxygen. The caveat is if you don't have oxygen, it's going to stop here and you're going to have to do fermentation. Unless you're an anaerobic respirator, then you're going to continue on doing that. Okay. This type of phosphorylation to make the 2 ATP is a direct taking from one molecule, putting it on the next, so substrate level. Okay, so I'm going to show you a little bit of what it looks like, as simplified as I can do, so that you don't get overwhelmed. Remember, I'm not asking you to memorize these steps. I just want you to see it a little bit, okay? So with the first 5 steps of the 10-step series of glycolysis, the first 5 steps are we're investing an ATP, and then we're investing another ATP. And you'll see what's happening. We put on phosphate group here, and then we put one on the other end, and that enables us to then split it in half to get our 3 carbon long molecule. And so this is the investment steps, or the preparatory stage. And then the second set of steps, the last 5 steps, we then get to get something back. So this is where we'll see our electron carrier NAD plus comes in and takes some hydrogen, and that's a bunch of potential energy now that goes to the electron transport chain. And we also take those 2 ADP that we just produced from here, right, 1 and 2, and those now take back phosphate. They become phosphorylized to give you 2 ATP. And then we do some more steps. We're going to have a little bit of water waste, and we're going to actually get another 2 ATP out of this. And now we have simple 3 carbons in a row. There's a little bit more going on. It's not just 3 carbons in a row. There's other atoms not being shown throughout this whole process, but just to get the main point across, this 3 carbon long molecule is called pyruvic acid, and you have 2 of them, remember, because you took glucose and split it in half. So each step, once you do the split right here, right, then every step happens twice because you have to do it to each molecule. And so this is where we we regain the 2 that we invested, and now we have a net profit of 2 ATP right here. And we also profit with this NADH potential energy. But this is only a profit if you're able to utilize it to harness that energy. So if you go to the electron transport chain, if it's running, then this is a profit. If you're not having if you're not really doing cellular respiration, but instead if you're having to do fermentation, then this is going to actually be a loss. But you need to keep regenerating the NAD plus. You have to keep doing this in order for glycolysis to happen. So you'll see the difference between fermentation and cell respiration. It's not just that you get 2 ATP from glycolysis, but you also get this NADH, which has a lot of potential energy that you only get to use. You only get to utilize that potential energy if you go to the electron transport chain. Okay, so NADH is a high energy molecule, and it's an electron carrier. Okay, next, some cells don't do glycolysis, and they use this completely alternate pathway here. And we see some examples like the Pseudomonas, rhizobacteria or agrobacteria, which are in the soil. Okay, and they're just doing this completely different pathway to use glucose. And instead of having NADA plus and NADH, they're using NADPH. Overall, it's the same idea as to get energy and to get ATP, right? And that's really kind of all we're going to say about that, is that this is an alternative pathway to glycolysis. I don't really care too much, except for that if we were trying to identify, right, an organism, this would definitely be a very helpful just to distinguish between these organisms and everything else. All right, so the pentose phosphate pathway is more important. This is happening simultaneously as you're carrying out glycolysis and getting energy from glucose that way. You also need to be able to have pentose sugars. These are 5-carbon sugars. These are the types of sugars that you find in your DNA, right? So you have ribose and you have deoxyribose, and those are pentose sugars, 5-carbon long sugars. And so we also take in a lot of sugars that are different from glucose, right? Different types of sugars. And so this allows us to break down some of those different types of sugars. And it produces a high energy molecule, NADPH. But it, as I mentioned, it's also supplying some building blocks that you need to make nucleic acid and amino acids, some of the amino acids. So you're going to see, in big picture, you're going to see this sitting on next to glycolysis happening simultaneously, so that we can utilize different types of sugars, and we can also produce different types of sugars that we need in the cell. Okay. So I've mentioned once you finish glycolysis, you have pyruvic acid, 2 of them, right? But we're not able to stick pyruvic acid into the Krebs cycle. Okay, and so here we're in the cytosol, we've made our pyruvic acid, and we want to go into the mitochondria and start the Krebs cycle. But once we get into the mitochondria matrix of the mitochondria, we have to decarboxylate, so remove the CO2, which is Highlighted in gray right here, C and 2 oxygen. So CO2 gets removed. Removing that is called decarboxylation, or to decarboxylate. Okay. And that's the sum of the CO2 that you exhale. This is where it's coming from. Okay. And then we also have to add, and so these are all different enzymes that are carrying out these reactions, but we also need to add coenzyme A, which remember we saw from when we were looking at different vitamins and cofactors and coenzymes, right? So coenzyme A gets added on to basically take the place of the carbon dioxide that we removed. And that molecule now is acetyl-CoA, and it's ready now for the Krebs cycle. And of course, you do this twice because you're going to have 2 pyruvic acid molecules. Pyruvate is another way to say pyruvic acid. It's slightly different. You see that it's you have this negative sign here. So now what's happened is the pyruvic acid acted as an acid and formed its conjugate base. So pyruvate is the conjugate base of pyruvic acid. I don't really care. I will use those words interchangeably because they can constantly go back and forth between the 2 forms. But it is technically the pyruvate that gets decarboxylated and turned into acetyl-CoA. I would not put on the test like pyruvic acid and pyruvate as 2 different options. I'd put one or the other. Okay, so the Krebs cycle, you have your acetyl-CoA and you're ready to do the Krebs cycle and then go into the electron transport chain. And all of this is happening in the mitochondria. So I want to show you, refresh your memory about the parts of the mitochondria and where these different processes are happening. So you're in the matrix of the mitochondria, which is shown here in blue. And that's where the Krebs cycle is happening. Okay. It's also called the citric acid cycle because the very first thing that gets made is citric acid. It's called the Krebs cycle after the scientists who discovered this process. Okay. So the 2 acetyl-CoA molecules go into the matrix, the blue stuff in this image. You're going to make even more carbon dioxide. So you're going to see another decarboxylation happen. You're also going to get out 6 NADH. Now this is counting for both of the acetyl-CoAs. This is the total for both. So if you're talking about one individual, it'd be 2, 3, and 1, and 1. But once both of the acetyl-CoAs go through, you get 4 carbon dioxide, 6 NADH, 2 FADH2, another type of electron carrier. So both of these are high energy molecules that are going to electron transport chain. And then you also get 2 ATP. And this is also made from substrate level phosphorylation. All of these electron carriers, the 6 NADH, the 2 FADH2, plus the other NADHs that we already made, all of those go to the electron transport chain, and we're going to get tons of ATP out of that. So let's briefly see where this decarboxylation and reductions are taking place. Again, I'm going to show you the steps of the Krebs cycle. I'm not asking you to memorize them. Oh, one more thing about terminology with the mitochondria. So the inner membrane, right? So mitochondria has 2 membranes. The outer membrane, which is shown here in darker brown, and the inner membrane, which is shown in like a tan or a beige color. And you notice that the inner membrane is folded. It's folded a bunch. Those folds are called cristae when you're talking about plural. And if you're talking about one individual fold, it's called a crista. And it's folded because that gives you a much more surface area, right? So if you were to take that inner membrane, take it out and stretch it out, it's going to be much longer than the outer membrane, right? So the folds create more surface area, and that means you can put more electron transport chains within the inner membrane. Okay, so the electron transport chain is embedded, the proteins of the chain are in the inner membrane. And by having the folds, you have a lot more inner membrane to put those proteins in, so you can have a lot more electron transport chains happening and produce more ATP at one time. And cells don't just have one mitochondrium, they have lots of mitochondria, right? And so lots and lots of energy being produced. Okay, so this is what the Krebs cycle looks like, simplified. But you put in your acetyl-CoA, you get citric acid, you carry out another enzyme, right? So you have an enzyme here taking acetyl-CoA and producing citric acid. You have another enzyme here that's going to reduce NAD plus to NADH. It's also going to do, you're going to have another enzyme that's going to decarboxylate, and you're going to get CO2. You're going to have another decarboxylation, get another CO2. You're also going to put in another NAD plus and get out NADH. And remember those NADH guys, high energy, potential energy going to the electron transport chain. And then here you put in your ADP, you get out ATP. So that's where ATP is made about halfway through the cycle. And then your other electron carrier that we haven't seen until now, FAD, comes in and picks up 2 hydrogens, becomes FADH2, right? So it's getting reduced because it's getting electrons, right? NADH is the reduced form. NAD plus is the oxidized form. So this is a redox reaction. This is a redox reaction, right? This is a redox reaction, and this is a redox. So you see you get 3 NADHs out of the whole single cycle. You get 1 FADH and 1 ATP. But that's just one acetyl-CoA. We have 2 of them for every single glucose molecule. So we're going to do this again. And that's how we get those totals that were on the previous slide. Okay? So that's it. I want you to remember what I typed on these slides, that the CO2 is waste. CO2 is produced from decarboxylation. You put in 2 acetyl-CoAs to the Krebs cycle, so your total products are the 4 CO2 waste, the 6 NADH and 2 FADH2, and those are electron carriers, high potential energy, and they go to the electron transport chain. And then you also will get 2 ATPs made, also from substrate level phosphorylation. This is all happening in the matrix of the mitochondria. Okay? Okay. The moment we've all been waiting for, the electron transport chain. To make lots of ATP, we're now looking at the inner mitochondrial membrane, and we're going to transport from the matrix through the membrane to the inner membrane space, so between the 2 membranes. And that's how we're going to see this whole process take place. So remember that prokaryotes don't have organelles. So when we're talking about bacteria doing respiration, they actually just have to do everything in their cytoplasm and then use their cell membrane, the plasma membrane, to do everything. Okay. So going back and thinking about eukaryotic organisms, we're going to study the electron transport chain as if it's a eukaryotic organism that we're studying, okay? Okay, so electron transport chain is a series of redox reactions where electrons are passed from one molecule to the next, and it'll be a higher energy molecule passing to a lower energy molecule, and this releases energy. And that energy is used to pump hydrogen protons. Notice the plus sign. These protons are pumped across the membrane. So you've gone from the matrix into the inner mitochondrial space. I have an image, don't worry. We'll take a look at this. But this pumping of protons only happens because of the redox reactions. The passing of electrons down the chain allow you to pump these protons, and pumping these protons traps them. The membrane is impermeable to H plus, to protons, and so you create a very steep gradient. And remember when we learned about transport across a membrane, when you create a really steep gradient, you have a lot of potential energy for it to flow back through to create equilibrium, right? But that membrane is impermeable to protons, so it can only diffuse back when a channel allows it to. And that channel is actually an ATP synthase. So it's actually also an enzyme, okay? And the enzyme will open up the channel and allow some of the protons to flow back through. And the energy that's released from that steep gradient and allowing a few protons back in is enough energy to power the synthase to force that third phosphate group onto ADP and produce ATP. Okay, so the ATP synthase is this powerful enzyme machine that produces ATP. Where did it get its energy? It got its energy from the electron transport chain. Passing those molecules down allowed you to pump those protons and make that steep gradient. And so this is the oxidative phosphorylation, right? We carried out all those redox reactions. So it was the oxidative phosphorylation. And we also use the term chemoosmotic because we made this really steep gradient of protons, which directly power the ATP synthase. Okay, so I know it's a lot when you go through metabolism the first time, sometimes even the second time, right? This might not be your first time seeing all of this, and you may still feel overwhelmed. I've tried to explain things as simply as possible, but it still takes a lot of repetition and a lot of grappling with this topic for it to become clear and not so overwhelming. Okay, so please do not feel bad if you are feeling overwhelmed. Remember to take your breaks. Remember to look at information in different ways, look at diagrams, read paragraphs, and ask me questions. Okay. All right, so excuse me for a sec. Okay, so I talked about the electrons getting passed from high energy to low energy molecules over and over again. So from protein to protein, eventually it needs to stop, right? And so you need to have a final electron acceptor. If you don't have a final electron acceptor, it's going to just find whatever is around for that proton to stick to, and that's going to damage things, right? So we need to have a final electron acceptor. Once you stop having that electron acceptor available, the whole system is going to shut down. The electron transport chain will shut down, and then the Krebs cycle will shut down. Okay, so you have to have a final electron acceptor in order to carry out cell respiration. If it's aerobic, the electron receptor is oxygen. If it's anaerobic, it's something else. Okay, most, if not all, eukaryotes are using aerobic respiration, and even a lot of bacteria also use aerobic respiration. It produces more ATP than anaerobic. Some bacteria do, however, use anaerobic respiration. Okay. And so as we see that electron passed over and over again, we get to pump those hydrogens across and make ATP. So this is, again, don't memorize all the specifics. I'm not going to test you on the names of the different molecules that you're passing the electrons to. I just want to demonstrate what I kept telling you, that NADH is going to go to the chain and FADH2 is going to go to the chain. And they're going to deliver those hydrogen that they picked up from glucose, and they're going to give it to the chain. And so then now you are oxidizing NADH to go to NAD+. So NAD+ is the oxidized form. It has this positive charge, right? Same with FADH2. You oxidize it, you take its hydrogen, which is, you're taking an electron, you're actually splitting it into, you're splitting the hydrogen from an electron and a proton. The electron gets passed down the chain, and the protons get pumped across, okay? And FAD is the oxidized form, right? So NADH reduced from previous steps, and then it gets oxidized again in the electron transport chain to NAD+, okay? All right, and then we see this happens over and over and over again, from the highest energy to the lowest energy, and then eventually letting oxygen be that final electron acceptor. You get a waste of water, right? So we've seen some water get produced throughout the whole process as waste. We also saw a lot of carbon dioxide waste, and so that should really help explain why when you breathe, you always exhale carbon dioxide. Typically, when someone suffocates, it's actually because they have too much carbon dioxide that they can't get rid of. They're breathing in too much carbon dioxide, right? Rather than what most people think of as not having enough oxygen. It's usually the other way around. Anyways, that's kind of morbid. We don't have to think about that. Okay, so again, don't memorize this chart, just memorize the concept that you have your electron carriers, NADH, FADH2, bringing in and donating those hydrogens, goes from high energy to low energy. You have your final electron acceptor at the end. You will have the electron from the hydrogen get passed, and then the proton that's left, a lot of those protons get pumped, but you also will have some protons that hang around, and they'll get used along with the oxygen to form that water as the final electron acceptor. Okay, I've given you some links here that you can use to see visual process from beginning to end. They're useful. I would suggest you check them out. Okay. And so this is just, again, to give you the overall idea of what's happening. So all of these proteins embedded, this is the electron transport chain. Some of these are pumps, right, to pump the proton through. And then some of them, like you see here, just this like disc looking guy, simply just passes the electron from one pump to the next. Okay, and again here we have another protein that passes the electron from this pump to this pump. And then finally at the end of the chain, you have your big muscle, right? Your ATP synthase. It's not actually muscle. I'm joking about that. Okay, but this is the guy that does all the work, right? He allows the protons to flow down that steep gradient, and by doing that, he gets a lot of energy that allows him to put that phosphate group onto ADP and produce ATP. And then that's also when you'll have a proton available to, in fact, to combine with oxygen and that electron. So the electron from the third pump does not get passed to ATP synthase. It stops at the third pump and will combine with oxygen and protons to make water. Okay? The ATP synthase simply is harnessing the energy that you have from all of the protons so that you can make ATP. Okay, where did all the protons come from? These pumps are pumping the protons from the matrix to the inner membrane space. Okay, and of course this is a eukaryotic organism because we're looking at a mitochondria. If it was a prokaryote, it just simply would not have a mitochondria, and it would just be doing this in its the only membrane that it has, the plasma membrane. Okay, and so by passing the electron, you get energy to pump protons. You make a very steep gradient, and that powers the ATP synthase. So hopefully you're feeling better about this process, and you're ready to move on. Okay, so cellular respiration yield in all of the steps combined for eukaryotes going through aerobic respiration, you will have a total net gain of ATP of 36. Okay, you have to spend some ATP to transport the pyruvate across the mitochondrial membrane. And so that's why you'll see that prokaryotes have a theoretical yield of 38 ATP because they don't have to expend energy to transport pyruvate. However, they don't necessarily get to make all 38 ATP because they are using their cell membrane, and there's no guarantee that they'll be able to have that steep gradient all the time because it's going outside of the cell, right? And they have to get it back in. So it's kind of complex, I understand. So we'll just go with these numbers, okay? But I don't want you to think that prokaryotes are always getting more ATP than eukaryotes. This is just like if in isolation of just individual cells when we study them, right? Okay, so anaerobic prokaryotes, they're going through respiration, but they're using something other than oxygen as the final electron acceptor. There are different final electron acceptors that can be used. So each pathway is going to give you a different number, and we're not going to worry about those numbers. But what we do want to know is that it's always less than using oxygen. Oxygen is always better. And so here's a summary for you. You have glycolysis is happening in the cytoplasm no matter who you are. The intermediate step to make acetyl-CoA also happens in the cytoplasm no matter who you are. The Krebs cycle happens in the mitochondrial matrix if you're a eukaryote and you have a mitochondria. If you're a prokaryote, it has to happen in the cytoplasm because that's all you got. And the electron transport chain, if you're a eukaryote, happens in the inner mitochondrial membrane. But if you're a prokaryote, you have to use the only membrane that you have, which is your plasma membrane. So hopefully you're starting to organize all this information, and it's becoming repetitive, I hope. And I've already mentioned all of this. Okay, so this is just repetition of what I've already said. And so let's look at those final electron acceptors, and then we'll call this part 2. And then we'll do a short third part on fermentation and biochemical assays, okay? So this is our last topic for this video. Okay, so when you have anaerobes, they could use something like nitrate, and they can have different pathways. So sometimes certain organisms produce nitrite, others have pathways to produce nitrous oxide, and others still have a pathway to go and all the way to make nitrogen gas. So I've put the molecule next to the name for you. We've already seen sulfate before, right, and hydrogen sulfide. And so we know there's some organisms that can use sulfate, but now we're talking about using sulfate as the final electron acceptor in respiration. It's a little different than what we were talking about when we looked at the SSA auger. Okay. So now the carbonate CO3 2- or carbon dioxide. So some organisms, usually archaea bacteria, can use carbon dioxide or carbonate and produce methane. And so the methanogens, the archaea bacteria. Okay, so good job. You have now learned about cellular respiration, and we will end here. We'll pick up here in the third part.